I would say the main challenge there is on probiotics and how they fit in the microbiome with regards to efficacy is mode of action, how probiotics might actually work. A lot of the studies don't address this. I suppose traditionally probiotics would have been selected based on their technological properties or ability to be able to grow them to high numbers and economically put them into, say, foods or supplements, but not necessarily for the health promoting properties. And I would say once we know how probiotics might work and how they might be selected, I think those sort of studies need to be done. And so I'm talking about mechanism of action. But I suppose when it comes to the whole microbiome, a lot of studies have in the microbiome, say, retrospectively look at disease. And in a lot of cases, look at shifts in microbiota composition that's associated with different diseases, be they like diabetes or autism, mental illness, whatever. But a major challenge is to show whether that relationship is just a response to the disease or may have some causal relationship to it. If it's causal, then they may be able to prevent the disease. However, if they're just a response, then what you're talking about is there may be a treatment to a side effect of the disease, but not going directly to the cause of the disease. I think it's only then, by understanding these mechanisms, that we will be able to understand how to optimize probiotics or optimize the construction of different microbial consortia that you can give to people to either treat or prevent the disease. Probiotics could be most effective in early life. And for a number of reasons. I think that in early life, it's a very dynamic time for the microbiota. If it's vaginally delivered birth, the infant has got the microbiota predominantly from the mother. And over time, that's kind of engrafted into the infant. And what you see is a lot of dynamic change in the first 6 months of life. And we believe there's a window there probably when you can change the microbiota in a positive fashion. One particular organism that's particularly dominant at that time, in Irish infants anyway, and in general, is bifidobacteria, and in particular, Longum infantis. And these organisms in early life can make up 20 to 50% of the microbiota. Preterm babies can be particularly a vulnerable population. They're born with, and the gut is not fully developed. They can succumb to infection, and necrotizing enterocolitis is a particular condition whereby they get infected, and it can be very, very serious. So much so that there have been a number of studies which have looked at probiotics in NEC, as it's called. And if you take all those studies together in a meta-analysis, it's fairly convincing that probiotics, even though different probiotics were used, and even at different different viabilities in the probiotic treatment, that they have efficacy. Some strains, and this is an area that we work on, produce a whole range of antimicrobials. They produce acids, but they also produce small peptides called bacteriocins. These are small. These can be highly potent. They're like antibiotics, except they're gene encoded by the organism. And these are small proteins that one bacteria produces to kill another one. We've basically mined the gut microbiota and have discovered a whole range of these. And so these are strains that are producing antimicrobials themselves. And what we're doing at the moment is we are looking at the ability of these strains to kill pathogens in the gut. And so this is one characteristic that we feel is a real probiotic trait of an organism. Its ability to produce antimicrobials to kill other ones. The gut is a very good competitive environment. And so some strains will produce antimicrobials to kill others. And so by targeting those strains and their ability to kill, certain pathogenic bacteria, I think we'll come up with a panel of probiotics for particular infections. I'm in a big microbiome centre in Ireland called APC Microbiome Ireland. It's quite a large centre of about 300 people. And we work in 4 different areas. One area is we work on microbes to molecules, we call it. And this is a discovery platform because we believe there are hidden secrets of mechanisms within the microbiome that we're only touching the surface of at the moment. I mentioned that we've screened basically hundreds of thousands of strains, looking for strains that are really potent at killing pathogens. And so we have a panel of those now. But one of the areas we're also working on is the virome. And so you have the microbiome, which is the bacteria, and you have the virome, which is the viruses. It's estimated that there's 5 viruses for every single bacterium in the microbiome. And we believe that this virome which a lot of it is composed of bacterial viruses, that they have a huge role in shaping the microbiome. And so a hot area in microbiome at the moment is looking at the virome. And so we've been isolating different viruses that hit key components of the microbiome. For instance, we just published a paper which, and this is with my collaborator Colin Hill and the group, where we isolated a phage called crassphage that kills Bacteroides intestinalis. And this is, these crassphage are probably the most abundant biological entities in the microbiome, but are only recently discovered. And so the microbiome still has a load of hidden secrets in it, and the virome is just one. But remember, these are viruses that are killing the bacteria in the microbiome, and so are having a huge effect. And so we're interested in this because I mentioned earlier that we're coming up with strains and peptides that kill things in the microbiome. There's also viruses that we're isolating that can kill particular strains in the microbiome. And we'd like to think that in the future we'll have, if you like, a toolbox whereby we can go in with precision biological tools where we can shape the microbiome in a particular way and not in a kind of a random collateral way like you have with antibiotics, where you just deplete the whole microbiome with a lot of broad spectrum antibiotics. But as we find out more about the microbiome, the bacteria in it, and how they work, we will be able to then hopefully go in and basically target those organisms, either put some in that are beneficial as regards probiotics and so on, but also take some out if we find that certain ones are undesirable in certain individuals. So I think you're going to see a lot of, as we find out more about the microbiome, as we find out more about the mechanism of action of the organisms, and also its causal relationships to disease, we will be developing precision biological tools whereby we can manipulate the microbiome in a certain way. And this will be not just probiotics, but also with antimicrobials, also with bacteriophage. Presently, one of the main, as we find out more about the microbiome, we're starting to find out, we already know the extent of it, and so it is a huge number of different species, and even a huge amount of strains within those species, thousands probably. The probiotics that are being commercialized to date are relatively few number, and they belong to certain genera, particularly Lactobacillus and Bifidobacteria. These bacteria, I believe, are really, really important. I mentioned Bifidobacteria in early life. However, they constitute only a small amount of the microbiota, of an adult, say. And so a big area in microbiome has been to culture the other organisms within the microbiome. It's proven very difficult in some cases because the microbiome has transformed microbiology. Microbiology as a discipline basically taught people like me how to culture organisms on their own. But in nature, bacteria exist usually in microbiomes, and they coexist with other organisms, and they in a nutritional ecological niche where they kind of feed off each other or they outcompete each other, and so they're used to growing with each other. And so when you try to grow them on their own, it can be quite difficult. As we find out more about the composition of the microbiome and how particular organisms within the microbiome have a direct effect on human health, one of the major limitations will be how do we culture those organisms in such a way that we can reintroduce them into people or increase the number of them. And there's two challenges in that. One is a lot of these bacteria are anaerobic and have very complex nutritional requirements. And so technically it can be very difficult, and also economically, they just mightn't be economically viable because it would cost too much to grow them. If you talked about the limitations of probiotics, I would say finding ones that are efficacious. They may be difficult to grow, and trying to find ways to cultivate them so they can be reintroduced to people or animals economically, and proving their safety and efficacy afterwards.